Joining us today. It's my pleasure to introduce Mike Miles, who's joining us today to talk about behavioral and expression genetics analysis of progressive ethanol consumption in diversity outbred mice. A little bit of background. I think most of us here probably know Mike Miles quite well, but he's an MD PhD, of course. He's currently working as both a neurologist and as a professor. He started out at, of course, in Stanford, and then going on to UCSF I cannot talk today, at the EGRC Center at Stanford, at UCSF. My brain is not working today. And then has moved from there to VCU, where he has rose to the ranks to full professor. And so he's been director of a training grant there for quite a while now, as author of many publications, and again, in a lot of areas of research that impact many of us in the society, and also has made just a great deal of contributions towards our understanding of especially ethanol responsive genes, ethanol responsive circuits, again, using techniques including BXDRIs, as well as diversity outbred, like he's going to tell you about today. And so with that said, I'm going to stop talking because I'm not doing well at it anyway, and I'm going to turn it over to Mike Miles.
Thank you very much, Chris. And I really appreciate the opportunity to talk to everyone today. I really appreciate the invitation. And it's good to see some people I haven't seen in a while. So, great. I don't know what is, okay. So I've been interested in the idea that genes regulated by alcohol may have something to do with the genes that regulate alcohol behaviors for quite some time. This slide was originally made, as says here, some 30 years ago, and I don't think PowerPoint even existed yet when I made that slide. But the idea was that we took the whole set of genes that were governing alcohol-related behaviors, and particularly at the time, we thought that some linkage studies and human genetic studies might be able to identify some of those. I've added model organism genetics as also could identify some of these genes, but we thought that ethanol responsive genes and networks of genes might also be able to help identify a subset of these genes involved in alcohol behaviors in humans. And so that's been our main way of going at things. But by combining expression genetics and behavioral genetics, that's really the way our laboratory works today. So we do a combined genetic analysis of ethanol behaviors and gene expression, for example, using mouse models. We have used the BXD and more recently the diversity outbred panels of mice for our genetic analysis. Because we're part of the VCU Alcohol Research Center, which is a cross-species analysis of genes involved in alcohol behaviors, we're able to take our mouse expression networks and gene expression data and co-analyze those with GWAS on alcohol use disorder or ethanol consumption, and also to with our colleagues Jill Bettinger, Andrew Davies, and Mike Grotewal, validate targets by cross-species higher throughput analysis with invertebrate models. And then we go about and rank these targets and test them directly through genetic modification or pharmacology in animal models, in our mouse models, and hopefully identify signaling mechanisms and circuitry underlying the behavioral responses. And also along this way, hopefully identify translational targets for alcohol use disorder therapeutics. As an example of some work that was done a little bit ago, we started out with BXD mice years ago, and we were studying the anxiolytic properties of alcohol. So alcohol is a great anxiolytic drug, and if anyone's ever studied anxiety in mice, alcohol is the best anxiolytic drug there is. Frankly, benzodiazepines show remarkable, strain variation and assay variation, whereas alcohol decreases anxiety almost in every assay and almost in every strain to more or less extent. So we mapped loci controlling alcohol's anxiolytic properties in the BXDs. Along the way, though, we did expression genetics across the BXD strains, and we're correlating that expression genetics data with our behavioral data to identify candidate genes for the anxiolytic properties of alcohol. I'm not going to talk about that today, but that's a whole nother line of very interesting work. But what we also generated were expression networks from this BXD expression data. And this is a small part of one of those networks. And all of these are, all the circles are genes, their relative ethanol response. Well, all of these were ethanol responsive genes. Their expression changed with alcohol exposure. And they were, in this part of the network, that there was a number of genes highly interconnected with other members of the network. And there was one gene in particular, GSK3 beta, that was highly connected. It was highly regulated by alcohol, and there was something known about its function in terms of synaptic plasticity that got us very interested. So we collaborated with our colleague Danielle Dick, who used some of the available COGA GWAS data at the time, and showed that when we took the GSK3 beta expression network, which is shown here, a very tightly correlated expression network of ethanol responsive genes, that using that network as a probe for association with alcohol use disorder, we found that the network itself was more significantly correlated or associated with that diagnosis than any single gene in the network. GSK3 beta alone had a significant effect as well. So furthermore, we did a variety of animal studies that I won't go through, that GSK3 beta overexpression in mouse prefrontal cortex increased ethanol consumption. Its deletion in CAMK2A neurons decreased ethanol consumption. And the selective pharmacological inhibitors also decreased ethanol consumption. A lot of this work is contained in this neuropsychopharmacology publication from a few years ago. But here on the next slide, I show you some of the recent, this is where we want to go eventually with all of these expression genetics and behavioral genetic studies. This is a highly selective inhibitor of GSK3 beta that's available for human clinical trials. It's actually in some human clinical trials for other disorders right now. And this is with the drinking in the dark model. If we look at baseline ethanol consumption over a 4-hour period, and then with titaglucid treatment, we see a remarkable decrease in ethanol consumption. And then if we wash out the drug over several days, the drinking behavior returns essentially to normal, and we can maintain, we can repeat this, et cetera. We've also used a constant access model of 24-hour access, and as well as an intermittent access model with 24-hour access. And titaglucid decreases ethanol consumption in all of those different models, but it has no effect on taste preference, locomotor activity, et cetera. So it looks like it's a promising potential drug for that could be explored in human clinical studies. So this has been our goal. But the BXD mice in the anxiety model were a little bit simple in certain ways. They were an inbred recombinant panel. We wanted something that was maybe a little closer to alcohol use disorder in humans. And for that, we needed a more complex mouse genetic model. So the diversity outbred mice, developed through efforts at many places, but UNC and Jackson Labs in particular, were really exactly what we were looking for. We studied the progenitor strains of the diversity outbred mice, which I'll show you in a moment, and they showed remarkable variation in their ethanol consumption using a model of intermittent access. But this panel of mice, as I'll say in a second, was developed for high resolution positional mapping of candidate genes. Furthermore, they are heterozygous at most locations, and they have a lot of genetic variation. So these are our goals in using the DO mice and studying a ethanol consumption related phenotype in them. And eventually, we hope to refine and validate targets using our cross-species analysis in gene targeting and pharmacology, as I mentioned earlier. So here's the DO mice. They're derived from these 8 strains. 3 of these were wild-derived strains. These 8 strains were predicted to contain about 90% of the total genetic diversity in the mouse genome. Each DO mouse from this outbreeding scheme is a unique, genetically unique individual. That's the downside of it. Well, in certain ways, we can't go get them off the shelf like we can a BXD recombinant inbred strain. We can't buy that same mouse from Jackson Labs, for example. So, but they are heterozygous, as I said, at most locations, and the recombination intervals are very small in terms of mouse models. They're not as small as in humans, but they're very small, allowing us to these linkage disequilibrium blocks allow us to have fine mapping of behavioral traits. So we used this DO mice in a study with over 600 ethanol consuming male mice. We wanted to do both, but that would have been, we required about 1,200 mice, and that was just not practical. We are studying females, though, in some follow-up studies for individual behaviors. So we had about 640 mice using a 3-bottle choice drinking model, 24-hour access every other day with water, 15% versus 30% ethanol. We use this sort of choice model because we found that some genetic manipulations alter the relative preference of certain of mice for a higher versus lower ethanol concentration without altering their total consumption. So those were some of the reasons why we chose this. They drank alcohol for about 4 weeks every other day, and then were sacrificed afterwards for tissue collection and genotyping. This is just to shows you here again the same scheme. This is the drinking period over these 4 weeks. Afterwards, we did a secondary anxiety assays, and then we collected tissue at this point once the drinking behavior had returned to its baseline after the secondary anxiety assay. And what you can see here is that this is a remarkable distribution of drinking behaviors across these mice, so that we have some mice at this end that are drinking, you know, between 15 and 40 grams per kilogram per 24 hours. Just a remarkable amount of intake in these, whereas we have others that are hardly touching alcohol at all. And these behaviors across these groups of mice tended to be stable over the 4 weeks of drinking behavior, although there were patterns of variation, where some mice started out drinking low amounts and then escalated, others started drinking higher and then decreased their consumption. And so we're analyzing that sort of microstructure of the behavior at the present time. It's actually very interesting. So this is the actual behavior. That's the subject of what I'll be talking about the rest of my talk here. This shows you the behavior analysis a bit more and displays what I was talking about in terms of some of the fine structure. If we just do a cluster analysis of the drinking days, we can see 2 large wings of this cluster. One contains the early drinking phase, and one contains the rest of the drinking days. And and you can see that there's maybe some substructure to this as well. But so this was very striking. You could see the mice that these drinking days were separated by no matter, we tried several different statistical approaches, multivariate approaches to analyze this data, and we came up basically with the same answer every time. So what we've chosen to do for a lot of our behavioral genetic analysis is to separate the first week of drinking from essentially the last week of drinking. So this later phase of drinking, and look at what genes and genetic loci are involved in this early drinking, the initial drinking phase versus the late drinking phase. And here is a plot of the whole population's behavior across the entire study. And you see the phenotype that we were hoping to capture, an escalation of ethanol consumption that occurred across the entire population, even though some mice, as I said, didn't escalate, but the entire population did increase their consumption. And if we look at the last week of drinking compared to the first week of drinking, there's a highly significant difference in their drinking behavior between those two time points, even though the behaviors are highly correlated with one another. So across the entire population, these are highly correlated, but they're also significantly different from one another. So this was exactly what we were hoping to capture, wide genetic and behavioral variation across the population with an escalation of alcohol consumption phenotype. So if this next slide is going to take a second to load, there we go. This gives you a sort of a busy summary of the behavioral QTLs, behavioral quantitative trait loci analysis. And again, we have the top row here is the first week of consumption. This is the last week of consumption. And these are interval maps going across all the chromosomes. So the LOD scores are on the y-axis, and the genomic position is on the x-axis. And we have three different behaviors mapped here. Two of them are related to one another. Ethanol consumption versus ethanol preference. And then the last one is 30% ethanol choice. So this is across the entire time frame, looking at the relative choice of 30% versus 15%. And just shown here, we have significant QTLs that were identified for all three of these behaviors. Here in the last week of ethanol consumption, there was a highly significant QTL on chromosome 4, whereas with ethanol preference, we found one on chromosome 13, and then with ethanol choice, there's a significant QTL on a chromosome 3. Down below here, I'm not going to go through this right now, but part of what we can do with the DO mice and because of we have a lot of information about the progenitor strains, and we were able to map out, with RQTL2, we're able to map out haplotype patterns so that we can see here that for the ethanol consumption on chromosome 4, that QTL, there's the prediction that if a mouse has an AJ allele in that location, they had lower drinking versus a mouse that has a NOD alleles or PWK alleles in that position, they would be drinking more alcohol, for example. So this is part of the power that we have with the DO mouse to be able to break this down into what we expect of the different alleles from the different progenitor strains. This is a summary table for all of our significant and suggestive QTLs. So we have more QTLs identified, but that are suggestive. The top significant QTLs are at the top of the table. You can see that in some cases we have multiple hits for across some of these behaviors. Here's last week ethanol consumption on chromosome 4. Last week ethanol preference, as would be suspected, is also, it's a suggestive QTL in basically the same location on chromosome 4, not quite as short of a interval. And then also for whole study ethanol consumption, across the whole study, this is just such a dominant QTL that's also significant in that with that phenotype. If you look here at these the support intervals for these QTLs, you can see exactly what we were hoping to find. Our support intervals are generally under, or in the vicinity of 1 to 2 megabases. There's some that are a little higher as we get to less significant LOD scores, but for these highly significant QTLs, they range between 1 and 3.8 megabases. If anyone's done QTL mapping in BXD strains or other mouse batteries or back crosses, et cetera, generally these support intervals are in the range of 20 to 40 megabases if you get a significant QTL. Obviously, with bigger panels, et cetera, those with the more extensive BXD panels now and et cetera, that those can be reduced as well. But this was, the initial DO mouse result. We had fairly narrow support intervals. This is the relative percent of the total variance in the phenotype that could be explained or that was attributed to these individual QTLs in this case. So in addition to the behavioral QTL analysis and positional analysis of candidate genes that we're going to get to in a second, I just want to mention here that we also, as I showed on the earlier diagram, we've also done RNA-seq analysis. To date, we've done it across 2 brain regions, medial prefrontal cortex, initially on about 220 samples, and more recently, in that analysis, it's still in progress. We've done nucleus accumbens across 400 drinking animals. And we've done 2 ways of analyzing this data, and this is again unique to the DO mice in certain ways. First was the traditional alignment to the C57 black 6 genome. That's, just like in human RNA-seq studies, you align to the generally to the consensus human genome. So we align to that reference genome, but also KB Choi at Jackson Labs has developed a program called GBRS, or genotyping by RNA sequencing, that allowed us to essentially align the RNA-seq data from each individual mouse to their progenitor strains, so that we could predict that in the region of a given gene for a given mouse, what's the genotype, what progenitor strain did it come from? And therefore we can improve our alignment and we can get essentially allele-specific expression, or at least an estimate of allele-specific expression using GBRS. And I'll just show, this is a powerful approach, and it showed that actually, when we just sort of a summary here, that if we use the alignment to C57 black 6, we aligned about 18,000 genes, whereas with GBRS, we increased that to about 22,000. So the overall alignment was much better. The number of cis expression QTLs, as defined by a LOD score just greater than 5 as an estimate, increased again by about 30%. And then the expression QTLs, which are potential strong candidate genes underlying our behavioral QTLs, increased as well within the support intervals of our 3 significant behavioral QTLs. And so you can see the number of cis QTLs and went from 50 to 65, et cetera. These overlap with one another extensively between the 2 alignments. But notice this one right here. This is our last week ethanol consumption confidence interval on chromosome 4, proximal chromosome 4. In the prefrontal cortex, we had a single gene with a significant expression QTL. And that expression QTL LOD score went up from the C57 alignment, about 15, to almost double, 28, with the LOD score using GBRS. So this is what we pretty much consistently see, not always, but fairly consistently. We see an increase in the LOD score, as well as the increase in the number of cis EQTLs that we identify using the GBRS alignment. But in this case, at least with the prefrontal cortex data, we essentially had reduced the genes in that interval to a single candidate gene. in one fell swoop. Now, I will, a little spoiler alert, when we've done our nucleus accumbens RNA-seq data, we actually there have a few other, three other candidate genes with cis-EQTLs, but this gene, CAR8, is still by far the strongest cis-EQTL in the support interval. Let's see here. This shows you a plot of the chromosome 4 support interval here in this panel A, and then some of the named genes that are in the support interval and their various transcripts that have been mapped for them. And you can see the CAR8 gene is right on the boundary, or on this proximal boundary of the support interval. There's several other genes that are in the interval here, but as I said, in the prefrontal cortex, none of these have cis expression QTLs, but we're not excluding them as candidates. But so there are other unnamed genes in this interval, but this fortunately, because of this narrow support interval, our number of candidates is fairly distinct. This shows you the cis expression QTL for CAR8 right here on proximal chromosome 4, and the haplotype pattern for CAR8 expression. And we're going to get into this in a minute. It's a little bit confusing to us right now, but very interesting, these haplotype patterns for the EQTLs. So you can see that the cis-EQTL for CAR8 is in strong linkage disequilibrium with the behavioral QTL, with 72 out of 79 markers overlapping, essentially. And there's a strong correlation between the expression of CAR8 in prefrontal cortex and the last week ethanol consumption. The correlations are not huge. They're about, but it's a negative correlation for both consumption and preference, and highly significant correlations, however, between CAR8 expression and the behavioral QTL result. The behavioral QTL looks very small here because of the strong cis-EQTL of CAR8. Here we show a comparison of the haplotype analysis for the behavioral EQTL from GBRS versus the behavioral QTL. And it's, we would love these to overlap with one another perfectly, but they don't do that. And if anybody else in the audience works with DO mice, I'd love to discuss this. But what we do see is here in the behavioral QTL, notice, as I said before, the AJ mice have a strong contribution in decreasing ethanol drinking, whereas I believe this is the NOD mice and the PWKs have a strong contribution. Alleles of those strains, of those progenitor strains, increase consumption. When we look at our GBRS expression QTL, the results are flipped, for one thing. So and that's so this AJ mice would have a higher expression of CAR8, and we've shown that that alleles from AJ mice increase CAR8 expression, and therefore greater CAR8 expression would decrease ethanol consumption, because as I showed, there's an inverse relationship between CAR8 expression and consumption. And so the AJs are doing the right thing. And here the PWKs are going the opposite direction. Their EQTL so would decrease CAR8 expression and therefore increase ethanol consumption over here. But the others are sort of, in the middle and sort of fuzzy. So there's not a perfect alignment across all 8 strains, but at least with 2 of the major contributors, we see them showing this this same kind of pattern that we would predict from the behavioral QTL. We've also done this in the nucleus accumbens, and there, I'm just not going to spend a lot of time on it. There seems to be different alleles that are jumping out at us, and they are not, but interestingly, even though we've got a strong cis expression QTL in nucleus accumbens, there's really no correlation between the distribution of these, at least for the major effects on gene expression of CAR8 with the haplotype pattern. So whether that implies that CAR8 expression in the nucleus accumbens is not that important for driving the ethanol behavior and that prefrontal cortex is more important, too early to say that, but that might be a suggestion. Here's just shows you, again, the CAR8 expression. Again, here as a validation experiment, we've jumped to a different battery of mice. We've gone back to the BXDs as sort of a genomic and bioinformatic validation of the DO mouse results. And here we've pulled phenotypes and gene expression data out of Rob Williams' gene network database, which we used all the time and have been using for over 20 years. Thanks, Rob. Here you see the behavior plotted on a y-axis, whereas the expression here are microarray data, expression of CAR8 on the x-axis, and this behavior is a ethanol condition place preference in the BXD mice. So this suggests that CAR8 expression varies inversely with a behavioral metric assessing alcohol's rewarding properties, perhaps, and therefore that might be consistent with CAR8's inverse correlation with ethanol consumption. Here in an ethanol sensitivity assay of ethanol-induced ataxia, we see a direct correlation between CAR8 expression and the behavioral phenotype. So again, possibly a consistent response that increased sensitivity to alcohol-induced ataxia might be expected to decrease consumption, for example. Here's just another plot of using a different phenotype from gene network. Here's CAR8 expression here, but in this case, I should have labeled this. In this case, the phenotype is drinking in the dark ethanol consumption. This was from the Williams Laboratory behavioral phenotype. Megan Mulligan contributed to some of this work, for example. And here we have a plot of the BXD strains and their CAR8 expression versus their drinking in the dark ethanol consumption. And again, we see an inverse correlation with CAR8 expression. So totally different genetic model than our DO mice, but a similar outcome in terms of CAR8 and ethanol consumption. And this was statistically significant, which I don't have the stats on there yet. So we've used this BXD data to do some modeling. And my student, Zach Tatum, has been working on this along with some of the bioinformatics core in our alcohol research center, to ask the question, can we model whether CAR8 expression is contributing causally to ethanol consumption in the DO mice? And so this is if we look at, and you'll have to ask Zach about all the details on this statistical approach. He explains them to me, but they sort of go in one ear and out the other for me, but I'm getting there. But that's what's supposed to happen, right? That as our students get into their third or fourth years, they're teaching the mentors, not the other way around. So here, if we just look at the contribution of genotype to the of the behavioral QTL, that it has a significant prediction of drinking in the dark ethanol consumption from this BXD data. Okay, that it can explain about 30% or so, 32% of the variance in ethanol consumption. But if we break that genotype contribution down to the contribution of the genotype to CAR8 expression, which is highly significant, and then we look at CAR8 expression effects on ethanol consumption. So here, there's a suggestion, because of the statistical significance of these wings, that CAR8 may be causal, that genetic variation, CAR8 expression may be causal in the genetic variation of ethanol consumption in this drinking in the dark data. So that's, a suggestion right now, but it's one more brick in the wall, as we like to say, suggesting that we should be looking at CAR8 more extensively. Anyway, this is just an early analysis of it, but a very interesting and exciting result using, again, a totally different genetic model and from the DO mice where a CAR8 was initially identified. So what's CAR8? Now that I've talked about it so much, what is it? What is it? It stands for carbonic anhydrase 8. So it's a member of the carbonic anhydrase family, but it actually has no enzymatic activity. It just has structural similarity to the gene family, and therefore it's a member of that family, but it does not have enzymatic activity, at least in terms of carbonic anhydrase activity. It interacts with the IP3 receptor and also PKC gamma and regulates receptor-stimulated calcium release. So it could have a very important role in neuronal excitability because of this. It's highly expressed in cerebellar Purkinje cells. There's been some suggestions that it's important for cerebellar Purkinje cell development, but it's also expressed strongly in cortex and dorsal root ganglia, but low levels of expression in non-neural tissues. There are existing human and mouse model genetics phenotypes that are interesting, that there's a null mutation that causes a wobbly phenotype in both humans and mice due to cerebellar dysfunction. And that's what I mentioned, that it's implicated CAR8 and possibly in cerebellar development, although some other studies have suggested that it's more cerebellar function and not necessarily development. Furthermore, interestingly, it's also been implicated in the genetics of analgesia to a number of different substances by QTL analysis in mice and some direct molecular studies. And we're actually studying its role potentially in alcohol analgesia as well as alcohol consumption. And as I've shown, it's regulated by alcohol acutely and with consumption. Well, I actually didn't show you this, but it is regulated in B6 or BXD mice. It's also regulated by amphetamine and cocaine in brains. So that's a little bit about it. We have currently a floxed mouse that we're starting studies on, and we'll know more hopefully in the near future. Taking us a step in a different direction or a related direction, we've also, as I've mentioned, we've done RNA-seq analysis across 2 different brain regions in our DO mice. And Maren Smith has initiated a detailed expression and expression network analysis in that data in the prefrontal cortex, and Zach Tatum is going to be slugging through the nucleus accumbens data. This is just an initial look at it. If we look at our high drinkers, if we take the 100 highest drinkers and we compare those to controls in DE-seq analysis, we get about 500 genes or so that are regulated by alcohol consumption, and with about a third of those going down and 2/3 of them going up in the high drinkers. Furthermore, Maren has done a WGCNA analysis looking at networks, because that's usually the mantra in our laboratory, that it's not the gene, stupid, it's the network that's important. And so here is a familiar heat map for WGCNA results. Here are WGCNA modules on the left on this y-axis versus on the x-axis, a variety of our phenotypes for first week versus last week, consumption or preference down here. And you can see the red colors indicates a strong positive correlation between the overall expression of a given module and that particular behavior. And this is done by taking the genes that are in one of these modules and generating a eigenvector or eigengene as that, which is the first principal component of the expression of all the genes in that network, and then correlating that eigenvector with your behavioral phenotype. So you can see these are highly significant correlations for last week ethanol consumption here for 4 different networks: red, turquoise, yellow, and salmon, for example. I'm not going to have time to go through more details of this. There's a lot of very interesting bioinformatics work that Marin's been doing on the genes within these networks and their possible regulators, as well as the eigenvector QTLs that are. Somebody could mute your. I don't know where that was coming from.
I think Stephanie, can you mute your microphone? Yeah, thank you.
There was a thunderstorm coming. So anyway, so this is going to be a very interesting analysis as we go on to identify these networks that hopefully are contributing significantly to these individual behavioral QTLs that we've identified. Also with our expression data, Zach has done just a whole genome-wide correlation analysis of expression versus our phenotypes, and here looking at last week ethanol consumption and the position of individual genes versus their log of their FDR associated p-value, we start to get an idea that there's a lot of genes that are showing highly significant correlations with ethanol consumption. And these may enlarge our catchment of genes that are important for these phenotypes beyond what we've identified with our behavioral QTLs. So this work and the expression network analysis is really very important. We will superimpose these types of correlation analyses and our differential expression analyses on those networks that Marin has analyzed, and identify networks that are overrepresented in genes that are correlating with our expression phenotypes. We would expect that these networks showing strong eigenvector correlations with this last week consumption, that a significant number of genes in that individual genes in that network would have high correlations with ethanol consumption. Furthermore, some of the genes are cropping up being interesting in and of themselves. This ZRAN-B2 has been contained in a couple of human GWAS studies on alcohol use disorder from the Million Vets analysis, and furthermore in the UK Biobank GWAS analysis, so that it shows some significance for ethanol consumption in those very large GWAS studies. So that just gets back to our cross-species analysis, where we were intersecting our DO mouse data and results with existing human genetic data, and then pushing some of our select genes into invertebrate models with our colleagues within the Alcohol Research Center. Here is an example of a further alignment of some of our results with human genetic data. And I'm sorry, I forgot to put the reference on this slide. This was a very great paper by Danielle Dick and colleagues. I believe it was in Nature Neuroscience about 1.5 years ago. And it was on an analysis of externalizing behaviors using essentially genomic structural equation modeling. And we're able to identify a fairly large number, about over 150 significant GWAS signals for externalizing behaviors, and alcohol maps onto externalizing behaviors. This shows a variety of these significant hits in Danielle's data that overlap with either our DO data or other alcohol expression data that's come out of our laboratory over the last 15 or 20 years. Some of that is acute ethanol responsive genes and BXD mice, for example. Others are, for example, work that Jim Bogenpaul and Sean Ferris and others did in Rhesus in collaboration with Kathy Grant's group, identifying networks, and Maren Smith was involved with this as well. Networks that correlate with ethanol consumption in a Rhesus model, something we published a few years ago, and as well as some human genetic studies. So we're looking at overlap, but notice the number of hits with our DO, ethanol correlated gene expression data, or our DO differentially expressed genes across these various GWAS hits from Danielle's work. And so I just highlight a few of these where we have multiple hits across these various data sources that we have. Some of these, CADM2, for example, has been identified in multiple GWAS for human alcohol use disorder and consumption. So this is encouraging us that our catchment of genes in the DO mouse, either by the behavioral QTLs or by expression correlations with these behaviors and differential expression, is going to be highly relevant to downstream analysis of human GWAS data. So with this, I'll start to wrap up here about talking about our conclusions. I think I've sort of tried to elaborate these as I've gone along, that we've identified significant behavioral QTLs with narrow confidence intervals, that it has increased our probability of identifying candidate genes much, much greater than anything that we'd ever dreamed about. For example, in those studies we started on ethanol anxiety with BXD strains 15 years ago or so. We've identified interestingly, there appears to be a couple of pieces of evidence suggesting there's something different about that first week of ethanol consumption versus this progressive phenotype of the last week. Both our principal component analysis and multivariate analysis of the behavioral data, but also the behavioral QTLs themselves, that the first week, the QTLs associated with the first week of drinking are distinct from those with last week. So again, suggesting different genetic influences on the initial ethanol seeking versus progressive ethanol consumption. We've been able to use a stepwise process to identify candidate genes. We identified CAR8 as a high priority candidate gene for this last week ethanol consumption due to its strong cis-QTL, its expression correlations with consumption, its haplotype pattern that has a partial overlap with the behavioral QTL haplotype. And furthermore, we've got other data from the BXD strains, as I've mentioned, that aren't contained on this summary slide. Furthermore, we've got this differential and the correlated expression patterns with our PFC and nucleus accumbens RNA-seq data that we believe is having impact on human GWAS studies. And furthermore, we have ongoing gene targeting studies that will eventually be used here in the near future to really try and validate some of these candidate genes and mechanisms underlying progressive ethanol consumption in the DO mice, when we hope that these have implications for human studies as well. We do have some limitations of this work, obviously. This was an all-male sample. As I said, we're going to be doing some follow-up studies on female mice, probably either in the collaborative cross strains or in some of the progenitor strains from the DOs. Our expression data is just bulk expression data from microdissected brain regions. It's not single cell sequencing data. But as of right now, single cell sequencing data just really couldn't get to the depth that we needed, but we will be exploring that in the future. We haven't validated any single candidate gene yet. We have a number of candidates though, but the number of candidates for some of the behavioral QTLs is going to complicate validation, as I mentioned here. Our high-ranking SNPs for these behavioral QTLs, in some cases, we're identifying genes for those SNPs, and that's something I sort of glossed over. When you do these analyses, what you're mapping initially is SNPs. You're mapping significance of SNPs for your behavior. You're not mapping the significance of genes per se. And most of those SNPs don't reside in the genes themselves, even in the introns. They reside next to the genes. And so we've only identified genes so far by positional approaches. We hope to use chromatin conformation analyses to really validate that a given SNP is affecting the expression, say, of the CAR8 gene. And then, despite all of our success, I think that the effect sizes and the polygenic contributions of alcohol consumption are going to make further direct validation of single genes difficult, particularly for chromosomes 3 and chromosome 12. But even for chromosome 4, it's possible that the CAR8 effect size, as we change its expression in discrete brain regions, is going to be small, and it's going to require a lot of mice to really confirm it. So, but we're highly encouraged, and we're going to proceed forward fearlessly. So, this is the group, and I'd like to thank everybody on the slide, really. Particularly today, I've talked about the remarkable work that was started by Kristen Mignogna and Lorna McLeod that initiated the DO mouse project, really. They slugged through and generated all the behavioral data on those initial 600 mice, and we have a lot of great stories from that heroic effort. Zach Tatum is a lot of the work I've presented today was from Zach's work. Zach's a fourth-year graduate student, PhD student in the lab. Maren Smith, as I've mentioned, is working on expression genetics, particularly with the prefrontal cortex data. But really the whole group contributes. And I'd like to thank my collaborators. I didn't mention this, but the drinking in the dark data was done by the Behavioral Core and the Alcohol Research Center, which is headed by Jennifer Wolstenholme, my long-term colleague, and the members of the Alcohol Research Center, particularly collaborations with Danielle Dick, with the externalizing GWAS, and also the COGA data that I mentioned early on have been particularly important. So I'll stop there and take questions.
Feel free to raise your hand if you'd like to ask a question, or you can type those into the chat and I can help them off. There were some earlier comments too, I'll point out. Richard Radcliffe pointed out that CAR8 is also cis-regulated in the L by S RIs. And then David Ashbrook also pointed out the VXVs. And I guess since I'm talking now, let's go ahead and take the first question here then too, which is I was very happy to see the AJ reducing effect, because I think our friends, the AJ mouse doesn't get a lot of screen time. And I was kind of happy to see those reducing alleles show up. And I guess I'm also curious though too about that reducing effect. Is that also known to be in other crosses like the C by A cross, for instance, or any of those other kinds of recombinant inbreds where AJ alleles are there?
So I'm sorry, can you repeat that? I was
yeah, sorry. Yeah. I was just mostly wondering about the reducing effect you see. I think it's the chromosome
AJs,
or the AJ haplotype there. And I'm just curious if that effect shows up in other RI crosses like that involve the AJ, like the C by A.
yeah,
or
I've looked for that, Chris, and finding ethanol consumption on significant numbers of other crosses involving AJ. If you have such data, I would love to see it, because I have looked for that, and I just didn't find enough of a large enough study to really generate a significant correlation for that. But AJ mice themselves don't drink much alcohol, as you know, so that's one thing we can say.
Right.
Let's see, where did the chat go? I think I closed it or something?
Also kind of interesting too that you don't actually see any, I don't think you showed any effects of the B6 alleles to actually increase intake, which of course is interesting then too. In this DO cross, you're not just simply not seeing those. And of course, those were the high drinking strains, which I think shows the advantage of looking at multiple crosses, of course, to get those allelic effects relative to other effects. Right.
I didn't mention the B6s, but the B6s tended, so see, this is the thing that's, if anybody's on that's a DO aficionado or working with DO mice, those haplotype analyses, what you see oftentimes, at least in our hands, you'll see 2 or 3 strains that show prominent peaks or effects, and then the rest of the strains will be like sort of in the middle. And so B6 mice tend to go in the same direction as the high drinkers, but they're sort of in the mud a little bit. That's what we've seen. So now the B6 alleles of CAR8 are they are very have different expressions. So they have a higher expression than either AJs or DBA2Js, for example. And so that is other data that other people have generated in studying CAR8 expression across various inbred strains. So their B6 mice would be a high, they would be a low CAR8 expressing line, consistent perhaps with their higher drinking. Obviously, there's many genes involved in that, but I think I've got that right anyway. Zach, if I'm wrong, tell me. But,
that sounds right. Can you hear me?
Yes.
Okay, yeah, that sounds right. And if you remember, the BXDs also showed up in the, or the B6s show up in the BXD data that we looked at as well, where they do fall sort of on the higher end of CAR8 expression, or higher end of ethanol consumption and lower end of CAR8 expression.
Right. Yeah, that one correlation graph I showed had B6 at the extreme of both of those. So David said something.
Did you see that in the chat? Yeah, David just posted.
If I can,
He just played with the BXD data, I'm guessing within Gene Network. And he says he has a chromosome 4 peak about from 3.5 to about 9.2 megabases, slightly bigger than the DO. And so it identified a similar QTL, but a larger confidence support interval for the log.
Now, that's for what phenotype, David? Is that for drinking in the dark, or?
Yes, ethanol consumption, drinking in the dark.
Right. Yeah, that was one of the phenotypes that I showed that Zach had made those plots of, and actually used one of those phenotypes for the mediation analysis. So yeah, it's a slightly larger interval, not too bad though for BXDs, and pretty darn good actually. And with those larger number of strains that are available now, it really helps. Although I don't know if that may not have been with a large number of strains.
38 BXD strains.
Yeah. So anyway, that's that's nice semi-confirmatory evidence. That's why I showed that BXD data. It was very interesting to see it adding some weight.
Yeah, I was just playing along at home.
Yeah.
Gene Network was great.
Gene Network, the number of hours I've spent playing on Gene Network over the last 20 years or so is just probably incalculable. Some of it though has resulted in figures and publications for sure. So, or at least some ideas that we've explored in it. So,
Other questions for Mike?
no other questions? Okay, well, thanks everyone again for listening, and Chris, I really appreciate the invitation. So it's nice to be able to tell this story a little bit. So
yeah, I was excited to hear about it. Thank you. It's a wonderful talk, by the way. So,
thanks.
All right, and thank you all for coming. And we'll see you hopefully in Galway.
Yeah, some of you, or at RSA.
Or RSA, yes. I got June. Thank you all. Have a good day.
Bye.